Allison, and we're here in the UC Davis Tahoe Science Center. And normally we welcome our doors to all of you, and we have all of these fun super sciencey experiments. But this year is a little different, as I'm sure you're all aware. But we're bringing it to you all virtually from our Tahoe Science Center. So our Tahoe Science Center is located here in Incline Village, and you can see the incline fault line right here. And this is the location where we normally start our Tahoe Science Center tour. And you can join us for our Tahoe Science Center tour on June 15th because we'll be reopening to the public. So we're super excited, and hopefully you'll come and visit us. So at the start, this is our watershed map. So I'm sure you all recognize this shape, very recognizable, the quintessential Tahoe, big, beautiful blue lake that I'm sure you all see every single day. But perhaps you don't notice a green area surrounding it. That green area is our watershed. So any precipitation that lands on this green area, whether it's snow that we're having right now, or rain or sleet, is going to ultimately end up in our common water body. And this is really important to understand because everybody lives in a watershed and everybody depends on fresh water in order to survive. And so it's really important that we take care of our watersheds in order to maintain our fresh, healthy drinking water. So this map behind me is also a clue as to this year's Science Expo theme. And it's all about Lake Tahoe. So not only are you going to see some of our favorite Science Expo activities, but you're also going to be able to learn a little bit more about this beautiful place that we call home. So I want to make sure that you all have your passport printed out. You're going to follow along with us and answer the questions along the way. And you'll notice that there's quite a few questions that start off with make a hypothesis, your educated guess. And we have boxes that you can check. You're going to check your guess before you see the experiment, and then you're going to check the correct answer to see once you actually see the experiment. And don't worry about being incorrect. Being a scientist is all about making mistakes, but learning from those mistakes. So we will together learn the correct answer. If you don't have it printed out, not a big deal. You can print it out later and you can quiz yourself to see how much you remember from our virtual Science Expo. And we want to encourage you, when we give you some time to make a hypothesis, to turn to your neighbor and actually have a discussion. Part of the scientific process is collaboration, tapping into each other's knowledge, what you already know, and seeing if you can figure out what the best possible answer would be. And if you have any questions, you can write them down, you can tell your teacher, and we'll be sure to try to answer all of them at the end of the session. All right, so I'm gonna check in with our amazing team and see if we're all ready to go, and it looks like we are. So we're gonna embark on our journey, starting on our research vessel. I'm gonna let Anne introduce herself, even though I just told you her name.
Oh, hi there, my name is Anne, and I'm here on our 2/3 scale model of our research vessel in our Tahoe Science Center in Incline Village, Nevada. And normally our researchers go out on this research vessel on the lake to take water samples and test those water samples and learn different properties of water and learn about the different properties of water. Today, we're going to be exploring some of those properties of water through 4 different activities that we have set up here together. We're going to start over here on the left side of our table, and we're going to explore something called density. For our first activity here, I have a jar of pure water from the tap here and a beaker full of vegetable oil. And in this experiment, I'll be pouring our oil into our water to see what happens and to see how these 2 substances and their densities interact with one another. So on our passport, following along, under the on the boat activities and question number 1, I want you all to make a hypothesis as to what you think will happen. You can put your hypothesis in the guess column. Do you all think that the water and oil are going to mix completely? Do you all think that the oil is going to float on top of the water? Or do you all think that the water is going to float on top of the oil? After we run the experiment, you can put your answer in the answer column, but I'll let you all think about it and discuss in your classrooms now. And while you all are making your hypothesis, I'm going to add some drops of food coloring to our oil to add a little bit of pizzazz to our experiment. All right, hopefully you all have had enough time to make a hypothesis here. In your classrooms, raise your hand if you think that the oil and water are going to mix completely. Okay, now raise your hand if you think that the oil is going to float on top of the water. A lot of people. And last hypothesis option, how many of you all think that the water will float on top of the oil? Some? Let's see, let's put our hypothesis to the test. You ready? Watch closely. Wow, our oil is floating on top of our water, and we can see that happens very, very quickly. The reason that this happens is because our oil is less dense than our water, and things that are less dense like to float on top of things that are more dense. But whoa, we can see some of those drops of food coloring are starting to sink down into the water. The reason that that is happening is because our food coloring is actually made up of water, and so it's more dense than our oil. After having a few seconds to sit there, the food coloring that's more dense sinks down through the oil and actually then sinks into the water, and we get to see these really beautiful color trails. Density, by definition, is just the amount of molecules or the amount of stuff packed into something in comparison to its size. So in this, our oil is less dense and has less molecules packed into it in comparison to our water. But this is maybe a more obvious experimentation with density, because oil and water are different substances. They're different things completely. But what if I told you that temperature can have an effect on the density of water? In this next experiment, I'm going to invite my helper Elise onto the stage, and we're going to fill our bathtub that we have here with 2 different temperatures of water. On the left, we'll have warm yellow water, and on the right, we'll have cold blue water. Elise and I are going to start filling the bathtub so you all can see what this will look like. We have a divider keeping these 2 temperatures of water separated right now. And to run our experiment, I'll be removing the divider so that we can make observations about what happens when these 2 different temperatures of water interact with one another. So looking at our hypothesis, our question number 3, or looking on our passport to make a hypothesis for question number 3, what do you all think will happen or hypothesize will happen when we remove our divider? Do you all think that the 2 temperatures of water are going to completely mix and make the color green, because when we mix yellow and blue, we get green? Do you all think that our warm yellow water will float on top of our cold blue water? Or do you all think that our cold blue water will float on top of our warm yellow water? I'll give you some time to make your hypothesis now and record it on your passport, and then we'll come back and run our experiment. All right, hopefully you've had enough time to make a hypothesis. What do you all think is going to happen? Raise your hand in your classrooms if you think that our colors and temperatures of water are going to mix completely and turn green. Some people think that. How many of you all think that our warm yellow water is going to float on top of our cold blue water? How many of you all think that our cold blue water is going to float on top of our warm yellow water? No one? So we have two main hypotheses that we're working with here. Let's take a look and see what happens when we remove our divider. And watch closely, because in your passport, you do have the opportunity to actually color in what you observe. Whoa! So we can immediately observe that as soon as we remove the divider, our cold blue water sinks down and almost looks like a wave as it moves underneath the water, underneath that warm yellow water. And as we've given them a few seconds to get settled here, we can see a very distinct layer of warm yellow water and a very distinct layer of cold blue water. Maybe we observe a little bit of green layers, the two colors are mixing. But for the most part, hold on, we're having some audio feedback. All right, but for the most part, we see the two very distinct colors and temperature layers. The reason that this is happening is just like in our oil and water, our warm water that started off on the left, our yellow water, is less dense than our cold water. And this is important in Lake Tahoe because Lake Tahoe's temperature changes throughout the year, at least the surface temperature changes. Maybe some of you have gotten in the lake in the summer and enjoyed it a little bit more because it's more pleasant than if you tried to get into it in the winter. I mean, we call it a polar plunge for a reason. If we look at this picture that I have here, each of these pictures represents the lake ecosystem or the lake water profile. And so with each picture, we see the changes throughout the seasons. In the winter, throughout the lake, it has a pretty uniform temperature. It's pretty cold all the way throughout. And that's because in the winter, our days are shorter. The sun is in the sky for a shorter time throughout the day, and we experience much colder temperatures. But as we move towards spring, we start to see longer days. The sun is in the sky higher throughout the day, and we start to get some warming of the surface of the lake. Just like you and I feel warm when we stand in the sun, those surface waters of Lake Tahoe can actually absorb that warmth as well and start to warm up over time. As we move to summer and we have even longer, warmer days, we see even more warming and maybe get even two different temperature warmth layers, as you can see in that image there or in that picture. But as we continue throughout the seasons, we move then back to fall and have shorter days again, and we start to see colder temperatures, and we start our cycle over again. And it's because warm water is less dense than cold water that when the surface warms up, that warm water actually stays floating at the surface of the lake and makes it much more pleasant for us to swim in it. In the summer, the surface of the lake can see temperatures between 65 and 70 degrees Fahrenheit, whereas in the winter, the surface of the lake is between 40 and 45 degrees Fahrenheit much of the time. So there's a big temperature difference just because of the seasons. And so if we look at question number 4 on our passport, which is actually on page 2 now, we have several different factors that are coming into play in Lake Tahoe. And question number 4 asks you what causes Lake Tahoe to form distinct layers or stratify. Stratification is the name for those distinct temperature layers. And what's causing this to happen is that we have warmer days in the summer and spring, and the surface water gets warmer, which creates a temperature difference between the surface of the lake and the deeper parts of the lake that don't receive that same energy from the sun. And as we just learned in our experiment, different temperatures of water have different densities. Just like our oil and water, that warm water floats on top of our cold water. So for question number 4, the correct answer is D, all of the above. All of those factors play a role in why Lake Tahoe sees stratification happening throughout the year. Moving on to our next experiment, we're going to learn more about another unique property of water. I have a glass of water here, just pure water from the tap, and I have a piece of paper that I've drawn 2 arrows on. And for this experiment, what we'll be doing is actually putting our piece of paper behind our glass of water and looking at the arrows through the glass to see how they appear to us. So for question number 5 on your passport, I want you all to make another hypothesis for this experiment. Do you all think that the arrows are going to stay pointing the same direction to the right? Do you all think they're going to change directions? Or do you think our arrows will appear to disappear as we look at them through our glass of water? I'll give you all some time to make your hypothesis now and mark it on your passport or talk to a classmate about it, and then we'll come back and we'll see what happens. Hopefully you all have had enough time to discuss in your classrooms. Raise your hand if you think that the arrows are going to stay pointing the same direction. Okay, now raise your hand if you think the arrows are going to appear to change directions. How many of you all think that the arrows are going to completely disappear? All right, so we have kind of a mixed bag here. Let's see what happens when we put our arrows behind our glass of water. Are you ready? They change directions, or at least they seem to our eye. Maybe you observe that they also seem a little bit more stretched out, maybe a little thinner and wider. I'll show you all the arrows in front of our glass again so you know I'm not trying to play a trick on you. And then we can see that they appear to change directions. And the reason why this is happening is because the light that is coming through our glass of water here is actually being bent. And so for question number 6 on our passport, the scientific word for the bending of light is something called refraction. And refraction, I have a diagram to help us understand what this looks like as our light is entering our glass of water. So like I said, when the light, whether it be coming from the sun or from a light bulb, comes into our glass of water, and we're looking at the arrows on the backside, the light is being bent. And so these 2 arrows, when on our piece of paper, look like they're pointing to the right. The light being bent through our water actually makes them appear as though they change directions to us. Refraction is a pretty unique property of water that can change the way that we see tons of different things, especially if we're looking into a lake. But there's another unique property of water that we're going to explore in this last experiment together that allows us to see why Lake Tahoe appears blue to us. For this last experiment, I'm going to actually have Tahoe Tessie help me out here. And Tahoe Tessie is our mythical creature that we love so much that lives in Lake Tahoe. In this experiment, I have a pitcher full of pure water again, and we're going to dunk our picture of Tahoe Tessie here that I put safely into a Ziploc bag so she doesn't get wet. We're going to put her into our pitcher and see what happens. So for question number 7 on your passport, this is going to be our last hypothesis here on the boat. Do you all think that when we put Tahoe Tessie into our pitcher, that nothing is going to change? Do you all hypothesize that her green color will disappear? Or do you think that Tahoe Tessie is going to completely seem to disappear off of our piece of paper? I'll let you discuss in your classrooms, and then we'll come back and we'll take a look at what happens when Tahoe Tessie swims. Okay. Raise your hand if you all think nothing is going to change here. Now raise your hand if you think the green color in Tahoe Tessie is going to disappear. And how about if anyone thinks she's going to completely disappear? No one? All right, let's put our hypothesis to the test. Are you ready? Watch closely. Whoa, her color disappears. So those of you that hypothesized that that green color would disappear were correct. But when I pull her back out, obviously that green color is still there. It's only when we put her into the water that the green appears to our eyes to disappear. This unique property of water that's going on has to do with something called reflection. And I have another diagram to help us see what this looks like. So the way that our eyes see color, and what you're going to get to explore with Elise in the 3D theater, is that light comes from the sun or from a light bulb, and certain colors will get reflected back to our eyes. So when we have Tahoe Tessie here, we are having green reflected back to our eyes. But water also does a really good job of reflecting light. And so when Tahoe Tessie goes into our pitcher of water, instead of that green color being able to reach the paper that she's on and be reflected back to our eye, that green never meets the piece of paper. So it seems like the green vanishes. Elise is going to go into much more depth on color and light with you all, because that's why Lake Tahoe actually seems so blue to us. But before we do that, I want us to make one more hypothesis here that we're going to circle back to at the end of our Science Expo together. I have 4 jars of sediment, and sediment is just another name for soil, or sometimes more commonly known as dirt. We have 4 different sizes, and they're scientific names of their sizes. We have gravel, which is our largest. We have sand, which is our second largest. Silt and clay are our smallest 2. And in these jars, I also just have water that we got from the lake. I'm going to shake up each of these jars, and I want you all to make a hypothesis in question number 8 on your passport as to which of these 4 sizes of sediment will take the longest to settle. And by settle, I just mean sink back to the bottom of our jar. We're going to learn about how sediments can change our lake ecosystem in our next activities, and we'll come back at the end of Science Expo to see if your hypothesis was correct. But I'm going to pass it off to Elise so you can learn about light and color.
Hey everybody, my name is Elise and I'm an AmeriCorps member here at Turk, and I am going to take you into the 3D theater because we are going to be exploring the properties of light, and this is a very dark room. And you might have seen our award-winning film Lake Tahoe in Depth in our Science Center before, but today we're going to be talking about the different colors of the way we perceive different colors. And we're going to start out with a way that we perceive color that I think most of us were introduced to color by. And scientists are introduced in color and the different wavelengths that colors produce, but artists are too. And maybe that is the first way that we encountered different colors. So we're going to start off with something that might be familiar with you. I have here 3 different colors of paint, and I have cyan, magenta, and yellow. C, M, and Y. And these are the 3 primary colors we're going to be mixing today. And you may have learned maybe back in kindergarten that the 3 primary colors are blue, red, and yellow. And those are just a simplified version that creates a bit of a darker color palette. So cyan, magenta, and yellow are often used by artists because they produce a really vibrant color spectrum. So I want you to take a look on your passport for question 10, which is on page 2. You're going to flip over. We have a blank Venn diagram that has these 3 same colors listed, and then a question mark right in the middle. And this question mark refers to what color will result when we mix the 3 colors of paint. So you have some options in your passport here. I want you to make a guess, make a hypothesis of what color you think will result when we mix these 3 colors. Will it be white, or will it be black or brown? So you can take a moment to discuss. Maybe you have some background knowledge into this particular experiment. Maybe you've even run this experiment at home or in art class. mixing all the colors together. I know I have. All right. So in your classrooms, I'd love for you to raise your hand if you think when we mix these three colors, we will get white. Anybody? Couple? All right. And who thinks that when we mix these three colors, we will get black? A few. Okay, what about brown? Also a few. So we have a bit of a mix. So let's see what will happen when we first mix in our magenta. And then we go to our yellow. I'm going to use a different paintbrush just to make sure these stay nice and crisp for you. All right, there's our yellow. And then what color do you see when we mix these two together? It's written on your passport at the top of our Venn diagram. This is kind of an orangey color, but it's also kind of red. So somewhere between orange and red. All right, and our last color, cyan, which is just a light bright blue. So when we have cyan over here, when we mix cyan and yellow, we get green, kind of like the middle of the stratification model that Anne showed you. And then when we mix our cyan and magenta, get kind of a dark bluish purple. All right, and all three, everybody's favorite part. What color do you notice? Yeah, so that's kind of a mucky brown color, right? And I would hope that when we mixed all of these colors, we would get a deep dark black, but instead we got a mucky brown color. So how are we seeing these colors at all? How do we perceive color? Well, that has to do with the way that light travels, and light travels in waves. So here I have the full spectrum of visible light. That's not all of the spectrum of light, but this is what the human eye can see. And the human eye is really good at seeing the colors red, green, and blue. We have special receptors in our eyes that pick up on the exact wavelength of those lights and translates them to our brain. But every single color has a different wavelength, a different size. And with white light reflecting off of them, we have white light coming from the light bulbs above our heads that are bouncing off of these different, of the paper in the background and reflecting back to you. So this white is the presence of all of these colors. White light contains all the colors of the rainbow. And then each of these different paints acts as a little sponge. And sponges are really good at absorption. So each of these different swatches of paint is absorbing certain wavelengths of light and reflecting or bouncing back other colors back to you. So for example, yellow is absorbing different colors, wavelengths, almost all the different colors of wavelengths of light except for yellow. And this specific wavelength of light is bouncing back to you. And so we have white light being absorbed, and then each one absorbs a different number of wavelengths and reflects colors back to you. So like I said, ideally, if we mixed all of these colors, we would get black. And here is a color wheel or a Venn diagram that we like to use that has those same colors we just talked about, but it is black in the middle. And I had mentioned CMY, and that might ring some bells for you. I know it did for me because I always think CMYK. And these are some printer cartridges that I took out of my printer. And we have CMY and K. So those four colors can be used by our printers to create all the colors of the rainbow when we print something out of a colored printer. And that's for the same reason artists use them, because they make really bright, vibrant colors. The K actually stands for key, but I remember it because it's the last letter of black. So we can explore how a printer can create all of these different colors. And I have here 4 different images of the Palace of Fine Arts in San Francisco. And each one of these images is using a different color from our printer cartridge. We have cyan, magenta, yellow, and black. And let's see what happens when we layer all of these together. All right, we have our cyan and magenta, making some darker blues. We add our yellow, Maybe we start to see some greens. And then finally, for detail, we'll add black. All right. What colors can you see? Shout them out in your classroom. What colors can you notice? You can be very specific. This is the time if you want to show off any fancy colors that you might know, maybe cerulean or chartreuse, perhaps. Okay, so first of all, I noticed this green color, which is our mix of yellow and cyan pigment. So we have green resulting in the middle. And then I noticed the sky kind of goes from a light sky blue, almost pure cyan, all the way to a deep dark blue and almost to an indigo. So that's pretty cool. So our printers are able to harness the power of pigment and absorb certain colors of light and reflect others back to you. And that's how I'm seeing this in this room. But how are you seeing this? Well, you are actually looking at a projection. You're looking at a computer screen, right? And so paints work by taking a white piece of paper, which is reflecting all colors of light back to you, and then we absorb some out. This is known as the subtractive system of light. Computer screens use an additive system because you start with a black screen, right? You start with it totally, when your computer is shut off, there is a black screen. And then we add colors of light to it. So let's start with a black room, a dark room, and then we're going to add the 3 colors an LED screen uses to reflect light back to you. And those colors are red, blue, and green. And so these colors are, remember when I mentioned that our eyes are really good at seeing 3 colors. They're good. They have receptors in our rods and cones for red, blue, and green. So that's why we have these 3 colors. And if you look on your passport, you have a second question that has a Venn diagram that has these colors in them. So we are going to be mixing these colors, and I want you to make a hypothesis again in your class about what color you think will result when we mix all 3 colors. So take a moment, debate, let your fellow scientists know what you're thinking, and you can use some prior knowledge or maybe we can do some research here. We have red and blue mixing, and then we have blue and green mixing. Red and green mixing. All right, let's put it to a vote. What do you think? Do you think when we mix these 3 colors, we will get white light? Raise your hand if you think we will get white. Yeah? All right, and raise your hand if you think, just like our paints, when we mix these 3, we'll get brown. No? Okay, well, let's see if you were right. 3, 2, 1. All right. You were right. So right there in the center, you may be able to notice that we have bright white light, which is pretty cool. And then something else that's super interesting is where our red light meets our blue light, we get magenta. When we mix our blue light and our green light, we get cyan. And then where our red light meets our green light, we get yellow. And this is the exact flip of the Venn diagram we looked at before. When we mix those colors of pigment, the overlap showed red, blue, and green. But when we mix the colors of light, our overlap shows magenta, cyan, and yellow. And then right in the middle, we get white light. So when we combine all these 3 colors, we get white light, which is how our LED screens are tricking our minds into seeing all the colors of the rainbow. Because we've kind of hacked our brain's wiring, which is really, really good at seeing blue, green, and red if we have all of our cones intact, which some of us don't, and that's what causes color blindness. That is how we can see the full spectrum of the rainbow. And so that's what happens when we add light, in the presence of light. But what happens in the shadows, in the deep, dark absence of light? Let's check it out. We're going to come right over here, and we're going to take a look at the 3 colors we just talked about. And I have a red light bulb, a green light bulb, and a blue light bulb. But you might not be able to see that, so I'm going to turn one of them on. Let's start with red. Okay, simple question for you. What color are you seeing on this background? What color do you see on this white piece of cardboard? Well, depending on your perception of light and perception of color, it might appear different to everybody, and that's totally okay. Me, personally, I'm seeing red. And that is because red light is reflecting from our light bulb onto our white piece of cardboard and back to my eyes and back to yours. So what else is going on here? We have a pencil that is propped up and I see a shadow. And shadows are just where light is being blocked. And remember I said that light moves in waves. So those waves move, even though they ripple back and forth in different sizes, they move in a straight line, just like the rays of the sun. So when our red light travels toward our pencil, at this exact moment, it gets blocked. It's being blocked by our pencil. And our pencil here is creating a shadow. And this is why we see this angle. So we have the red light on the left, the pencil in the middle, and the shadow to the right. Okay, let's see what happens. Keep your eyes on that shadow as I move to the green light. What do you notice? Okay, first of all, I noticed that this background is appearing green and the shadow jumped a little bit. It's now directly behind the pencil, which makes sense because the light bulb is directly in front of the pencil. And watch that shadow as we turn on our final light bulb alone. Okay, so now we have a blue background. We have a light bulb, a blue light bulb on the right, and that light is traveling toward our pencil. It's being blocked and creating a shadow to the left. So you don't have any passport questions about this particular activity, but I do want you to make a hypothesis using the information we just learned with our moving flashlights about what you think will happen when I turn on a second light bulb. I will be turning on the blue light bulb and the red light bulb. So blue light and red light. I want you to make a guess, take a chat with your neighbor about what color you think the background will look and how many shadows you believe will appear. So take a moment. What color do you think? And this is, again, just out loud. What color do you think the background will be? How many shadows are we going to get? And for my sake, I would love for you to hold up fingers to let us know how many shadows you think you'll see. 1, 2, 3, 5, 8. Most folks are saying 2. All right. And what color do we think? Do we have a consensus on color? Let's see. Well, when we held up our 2 flashlights, you can take a look at that Venn diagram you had before. When we held up the red flashlight and the blue flashlight, where they overlapped, it created magenta. Isn't that beautiful? So we have red light and blue light combining to create magenta. And you were correct. We do have 2 shadows, but they're not gray anymore. These are not some boring shadows. These are very interesting colored shadows. How is that happening? Well, our shadow on our left is being created by our blue light bulb. I can prove it to you if I cover up this blue light bulb. Our red appearing shadow goes away. And so this shadow is being created because the blue wavelength is moving toward the pencil. It's being blocked and bounced back. And in the absence of blue light, our red light comes to the rescue and fills it in. The same thing is happening in reverse over here. So we have our red light traveling from the light bulb toward the pencil. It's being blocked. It's being blocked. And in the absence of red light, we have blue light coming in to join it. Okay, I'm going to turn on our final light bulb. I want you to take another guess. How many shadows are we going to see? And what color will the background be? If you're feeling extra excited about this, maybe you can take a guess as to what color our shadows will be. So just take a few seconds, chat with a partner. All right, let's check it out. Look at that. So our white piece of cardboard is finally looking white to me. And that is because we have red, green, and blue. At the center of our Venn diagram, remember, we had white light. So these 3 colors are combining to create white light. And then in our shadows, we have cyan, magenta, and yellow. And those 3 colors just keep on coming back to us. Why are we seeing these 3 colors again? Well, if you take a look at our shadows in order from right to left, we have, first of all, we have a cyan shadow, and that is being created by this light bulb, right? Our red light bulb to the left is creating a cyan shadow to the right. And I can prove it again. If I cover this red light bulb, the shadow disappears. So this is the shadow that's created by the red light bulb. And in the absence of red light, our green light and blue light combine to make cyan. The same is happening here where the green is blocked, our blue and red combine to make magenta. And here where our blue light is blocked, we have red and green combining to make yellow, which is pretty cool. Who would have thought that if you combine red and green, you would get yellow? And I wish you all were here to make some shadow puppets with me, because it is pretty awesome to just see how all of those different colors of light combine. And so this is how our colors all combine together to create white light. But I want to prove to you that we can take white light and create all the different colors in reverse. So let's take a look at our next experiment. So I have here a white flashlight, and our white flashlight is created, white light is created by all the colors of the rainbow. And so like in a rainstorm where we have raindrops that are diffracting out the different colors, here we have a prism. And this prism will separate out all the colors of the rainbow from one white light bulb. Can you see them? They're moving all around. Pretty cool, huh? So, but what does any of this have to do with like Tahoe, right? This is all about Tahoe. Well, when I think color and when I think Tahoe, I think blue. And that is probably because of how many bumper stickers I've seen in my time. And Lake Tahoe is blue for a lot of different reasons. And question 14 asks you about why Lake Tahoe is so blue. And Lake Tahoe is blue for a number of reasons. First of all, it's the properties of light. And so we have here an image of the sun, which I just said, white light contains all the colors of the rainbow. And so when white light hits the water, it is separated out into all the different colors that create it: red, orange, yellow, green, blue, indigo, and violet. And you have an image that looks very similar to this on page 4 of your passport. And page 4 of your passport asks you to circle which colors get absorbed by the water first. So every different color has this different wavelength, and red, orange, yellow, and green get absorbed by the water first. So you can circle those ones, and those get absorbed as heat. And so the wavelength is just too big, too slow to reach the bottom of the lake. It gets trapped by bumping into different water molecules early in the lake, and that is why Lake Tahoe is ever even warm enough to dip a toe into. But what about blue? Well, blue is the perfect Goldilocks shape wavelength to reach through, dodge past those water molecules and any of the potential sediments or algae we might find, reach the bottom of the lake and reflect back to our eyes. So blue, indigo, and violet. You have another question at the very bottom of page 3 of your passport. I know we're flipping back and forth, but those 3 colors are reflected back to our eyes. And that is why Lake Tahoe looks so blue. But those are not the only reasons Lake Tahoe is blue. Yes, it is the properties of light, but there are a couple other things too. Have you ever seen a lake less blue than Lake Tahoe? I have. Most lakes I've seen are less blue than Lake Tahoe. What are some other color lakes I've seen? Well, I've seen some that are pretty brown, and I've seen some that are pretty green. What do you think is causing those different colors? You have a hint in your passport. So question 14 names a couple different reasons why Lake Tahoe would appear less blue. One of those is an increase in sediments. So if there were a lot of sediments in Lake Tahoe, it might appear like those jars that Anne shook up, brown. Just like when we mixed all of the colors of paints together, we created a brown color. The same thing would happen if we increased a lot of sediments into Lake Tahoe. In fact, some of the first paints were made from different soils and different sediments. And so that is one reason Lake Tahoe could appear less blue. Another reason would be if we introduced a lot of nutrients, like phosphorus or nitrogen. Those are chemical fertilizers that farmers would use to help their plants grow. So phosphorus and nitrogen are really good at helping plants grow. And plants on land are often, what color? Green, right? So why are plants green? I've never even thought to ask that. Well, it's because their chlorophyll absorbs red light and blue light and reflects green light. So just for the same reason that anything appears a different color, plants are green. And when we include those sorts of fertilizers or nutrients to a lake, it also helps plants grow. But in a lake, we don't have corn growing, right? We have tiny, tiny particles of algae. And algae are just tiny floating plants in any body of water. And when we have an influx of nutrients, those plants will increase. And that would make our lake look more green. So because we don't have very many sediments in Lake Tahoe, because it's very clear and deep, our light can reflect back. And because we have very few plants like algae that grow because of nutrients, our lake looks very blue. And so the answer to question 14 is all of the above. So this is not only important because Lake Tahoe is beautiful. I mean, I really like that it's blue and clear, but this is also important for the organisms that live in Lake Tahoe. So I'm going to pass things off to Bailey, and she's going to explore some of the different organisms that you have that we might notice in Lake Tahoe. So you can turn your passport to page 4 to In the Lab, and Bailey is going to take us on a tour of our fish lab.
Awesome. Thank you, Elise. So my name is Bailey, and I am, as Elise was saying, going to take us on a tour of our Tahoe Science Center Fish Lab. So you can follow me out of our 3D theater. We're going to step back into the lights and into our fish lab. So over here we have a variety of aquariums that are home to a few different aquatic organisms that you can find in Lake Tahoe. And aquatic organisms are just animals that live in the lake. And some of them are native, which means that they are from Lake Tahoe. They've always been here. And some are non-native, which means they're actually from a different lake or a different body of water, and they were introduced into Lake Tahoe either on accident or on purpose. And we're going to explore this with each organism that we look at today. So we can start over here with our Tahoe Science Center favorite pet. Her name is Lonnie, and she is a Lahoten cutthroat trout, which is the only native trout species to Lake Tahoe. If you've heard of other trout, so like our brown trout, brook trout, or lake trout, those are all non-native trout species that were introduced into Lake Tahoe on purpose by scientists so that there would be a wider variety of trout for fishermen to catch. And actually for a while, scientists thought that Lahontan cutthroat trout actually were extinct, which means that there were no more left in Lake Tahoe or any of the other lakes in the Sierra that they're native to. But as we can see, because we have Lonnie here, that wasn't the case. So scientists over on the border of Utah and Nevada, so a whole state away, actually found some fish that looked really similar to Lonnie here, and they turned out to be Lahontan cutthroat trout, so they were not extinct. And they were able to use those fish and their eggs to bring back the Lahontan cutthroat trout populations in the lakes that they're native to in the Sierra, like Lake Tahoe. And one of the ways that we can help do this is actually through a program called Trout in the Classroom, where we go to a local fishery and we get some Lahontan cutthroat trout eggs and we deliver them to classrooms so that students like you can help us raise these eggs into baby fish, also called fry, and then we can release those fry into streams and creeks that flow into Lake Tahoe so that we can bring Lahontan cutthroat trout back into our lake. And this program is going on right now. So right behind me, we have our Lahontan cutthroat trout nursery. And if we can zoom in, we can hopefully show off some of the fry that we have raised. Now they're really hard to see, so you're gonna have to look closely. These fry are only about an inch long, so they're very tiny. Once they grow to longer than 3 inches, they will enter their next life cycle stage, which is a fingerling. And then after that, they will slowly grow up to be adult, mature Lahontan cutthroat trout like Lonnie. So we can come back over to Lonnie. And we actually raised Lonnie from an egg 5 years ago in this same program. So Lonnie is 5 years old. And Lahontan cutthroat trout in Lake Tahoe would normally eat smaller fish species and a variety of zooplankton, like Meister Shrimp and Daphnia, which are the other 2 organisms that we're going to take a look at in just a minute. So what I want you to do is we're going to get up close and personal with Lonnie as I feed her some of those Meister Shrimp that I was just talking about, which are a non-native species to Lake Tahoe. And I want you to make some observations on how you think she's finding her food. So do you think that she is using her nose to smell her food and catch it? Or is she using her eyes to see those tiny shrimp and catch them and eat them? Or do you think Lonnie has some other way, some other mechanism of finding her food in the water? Give her another bite. Oops. So I don't know if you can tell, but Lonnie is actually using her eyes to find her food. So this makes her a visual predator. She needs to be able to see her food in order to catch it and eat it. And that is true for Lahontan cutthroat trout and many other fish species in the lake. They rely on their eyes to see and find their food, which is one of many reasons why it's important that we keep Lake Tahoe clean and clear of those sediments and algae so that our fish can more easily find their food. But let's actually transition over to looking at the food that I was just feeding her. So if we take a look at this petri dish right here, this is a Meister Shrimp. You can use my fingernail for scale. It's about somewhere between 1/2 inch to 1 inch long, and they are a non-native zooplankton. So zooplankton, zoo is a Latin word for animal, and plankton means something that's really, really small and lives in the water. So that just means that this is a very small aquatic animal. And since it's so small, it can be kind of hard to see on our camera, I'm actually going to put it under our microscope here, which is attached to this TV screen. That way we can all take a look at it up close. Okay, so this is our Meister Shrimp. And as I was saying, this is a non-native species to Lake Tahoe. So it's not originally from our lake. Scientists actually introduced Meister Shrimp on purpose so that they could act as a food source for our fish, like Lonnie. We saw how much she liked eating them. That way our fish in the lake would actually grow bigger, so fishermen could catch larger fish. Unfortunately, there's a few things about Meister Shrimp in Lake Tahoe that scientists didn't quite understand or predict, and it led to some unintended consequences. So they didn't end up serving as a very good food source for our fish. And if we take a look at our mysis and make some observations, we can start to understand why. So when I look at our mysis, one of the first things I notice is actually these dark spots at the front of the mysis's head. And those are their eyes. So they have really large eyes relative to their whole body. And because they're so large, they're actually really sensitive to sunlight. So when mysis are swimming around in those clean, clear water in Lake Tahoe, the sunlight is penetrating the water easily, and it's really painful on their eyes, those harsh sun rays. So mysis, to avoid the sunlight, are actually swimming down to the bottom of Lake Tahoe and hiding in the deeper, darker parts of the lake. And if you don't know, Lake Tahoe is a really deep lake, and on average, it's about 1,000 feet deep. So our mysis shrimp are swimming 1,000 feet down to the bottom of the lake and hiding in the dark parts during the daytime. And as we know, we just saw with Lonnie, the fish in Lake Tahoe are relying on their eyes to see their prey. So if mysis, which are supposed to be their food, are hiding in the dark, our fish can't really find them. So not actually serving as a food source for our fish. And instead of our fish getting bigger, they've actually gotten smaller because not only are they not a food source, but they're also, the Meister shrimp are also eating the food that the fish would eat without the mysis. So that's our Daphnia. So after the sun has gone down, our mysis actually swim back up to the top of the lake. So they swim another 1,000 feet, and they have to eat a lot of food to fuel that swim. So when they come back up to the top of the lake, they're eating algae, those tiny aquatic plants that we were talking about. And they're also eating Daphnia, which is a native zooplankton that we're going to look at next, that normally our Lahontan cutthroat trout would eat as well. So they're not serving as a food source for our fish, and they're eating up the Daphnia so that there's not as much for our fish to eat, which is causing the fish in Lake Tahoe to get smaller. And it's also, they've eaten so many Daphnia in the lake that they're actually depleting the Daphnia populations, which means that there's not a lot left in Lake Tahoe, which has actually had an unintended consequence on the water itself. So because there's no more, or there's not as many Daphnia left in Lake Tahoe, lake clarity has actually gotten worse. So it's not as clear as it used to be. But to understand why, let's take a look at Daphnia. So Daphnia is a native zooplankton to Lake Tahoe, and I have one here that's swimming around in this little drop of water. So this one is alive, and it's that little dot that you might be able to see moving around in this drop of water. So Daphnia are significantly smaller than our Meister shrimp. I'm going to put this under our microscope. And this little circle right here that's moving around, that is our Daphnia. So I haven't switched any of the settings on our microscope yet, so it zoomed in the same amount. And we saw that the mysis actually took up most of the screen, whereas the Daphnia only take up a really small amount. So we can see how much smaller our Daphnia is than our mysis, which is how mysis are able to eat them. But if we zoom in so that we can take a closer look, be a little less because this guy wants to move around. Okay, so one of the first things that I notice or observe about our Daphnia is that it has a transparent body, which means that it is see-through. Might have to zoom out a little bit. So this line that goes down the center of our Daphnia's body, that's actually their digestive system. So we can see inside the Daphnia, which actually makes it really easy for them to camouflage themselves in the clean, clear water of Lake Tahoe. So they blend in with the water really well, which is supposed to help them hide from predators like Meister Shrimp and Lahoten cutthroat trout. Another thing that I observe, get this a little more central, is these things that are moving around, they're kind of twitching out of the top of the Daphnia. And those are actually antennae. So there's two antennae that are sticking out of the top of the Daphnia's head, and that is how it moves around. So that's how it's swimming. It's also how it feeds. So Daphnia are filter feeders, which means they're filtering their food out of the water. They're not being very picky. They're not hunting for their food. They're actually using their antenna to push water into their mouths, and they're just eating anything that's small enough to fit inside their mouth. So that includes algae, those tiny aquatic plants, and it also includes fine particles or sediments like the ones that Anne shook up in the jars over on the boat. And only the two smallest ones, silt and clay, can actually fit inside our Daphnia's mouth. So all of these little dots that we see floating around, oh, looks like she was almost about to eat one. Those are different small fine particles or sediment that if we left the Daphnia in this drop of water for long enough would actually eventually get eaten by the Daphnia, so filtered into her mouth, and then it would travel in through its digestive system and come back out as a more densely packed fecal pellet. And as we learned on the boat with Anne, things with greater density will sink, and it will sink down to the bottom of Lake Tahoe and actually help improve lake clarity. So just by eating the way that they naturally do, they're actually cleaning up the lake. They are serving as a little vacuum cleaner in Lake Tahoe, which is pretty cool. But since we know that mysis are eating so many Daphnia and there's not that many left in Lake Tahoe, this has actually had an effect on the clarity of the lake. So because we're removing all of those vacuum cleaners, or Meister Shrimp are removing all of those natural vacuum cleaners, lake clarity is decreasing. It's becoming less clear. So it's another thing that scientists didn't really anticipate when they introduced Meister Shrimp as a food source. So we saw how the introduction of just one non-native species, the Meister Shrimp, has influenced two of our native species, the Lahontan cutthroat trout and the Daphnia, by not serving as a food source and eating up all of the food for our fish so that our fish are getting smaller, and eating up all the Daphnia so that their populations are crashing and the lake is decreasing in clarity because we don't have as many natural vacuum cleaners. So we're going to take a look at the last page of our passport here. And we are in the lab, so we're on question 18. So are Lahontan cutthroat trout native or non-native? Well, we learned that Lahontan cutthroat trout are in fact the only native trout species to Lake Tahoe. So you can circle the word native. And on question 19, are Meister Shrimp native or non-native? These are in fact non-native. They were introduced on purpose as a food source, even though we know that it didn't really work out that way. Question 20, Meister Shrimp hide in the depths or the shallows of Lake Tahoe where fish can't find them. We learned that they are actually swimming 1,000 feet down to the depths of Lake Tahoe to avoid the sunlight. Question 21, are Daphnia native or non-native? They are in fact native. They're the native zooplankton. Question 22, true or false? Daphnia can eat algae and fine sediments and help the lake become more clear. This is true, so by being filter feeders and filtering out those fine sediments, they are improving lake clarity. And then question 23, our final question of our Science Expo passport, is true or false? Lake Tahoe's food web has not been altered by non-native species introductions. So a food web is how all of the aquatic organisms in Lake Tahoe are connected based on who eats who. And so we saw that the introduction of just one non-native species, our Meister Shrimp, has affected 2 of our native species, and possibly even more that we didn't get a chance to talk about today. So this is false. They have, non-native species have affected our native food web. And Meister Shrimp are not the only non-native species in Lake Tahoe, but that is all the time that we have to talk about. So I am actually going to not run into this poster. I'm going to bring this back over to Anne, who is in front of the boat with our jars of sediment that we shook up at the beginning of our Science Expo. And let's take a look at what they look like now.
All right, so we can see pretty quickly looking at our 4 different sizes of sediment that we shook up before we went into the 3D theater that our silt and clay are taking the longest to settle or sink back to the bottom. And this has to do with the fact that larger sediments like gravel, as you can see here just looking at them, they have more mass, they're bigger. They're heavier, so they sink down in the water really easily, whereas the clay and silt are much smaller and don't sink down as easily. And as we've gotten to explore on our Science Expo tour of our Science Center today, when we have sediments in the lake, we can actually change the color that the lake is appearing to us. It might appear far more brown than that beautiful blue that we know and love so much. But also, as all of those different organisms that Bailey just introduced you to, because our food web has changed, because Daphnia populations are much smaller than they were initially in Lake Tahoe, sediments are having a harder time even now without the Daphnia populations to sink down to the bottom. So there's a lot of different forces and factors that go into everything that happens in Lake Tahoe. And we could only share that small bit of information with you today, but we're so glad you were able to join us for our Science Expo activities. And I'm going to pass it back to Allison so that we can field any questions you all may have.
Okay, so it is 12:07 right now. So I know that we've gone a little bit past our initial time estimate, but if you have a little bit of flexibility in your time, we would love to take any questions you have. You can either unmute yourselves and just have the students say the question out loud, or you can put it in the chat, whichever is more convenient to you. I thought I saw a hand raised in Mr. Henderson's class.
So is there any way that we can help?
Oh, I might need, hold on a second. I might need to raise the volume up a little bit. Yep, I shut that off. All right, can you ask that one more time?
Is there any way that we can help like making little robot things of those vacuum cleaner things?
Oh, you're talking about robotic Daphnia?
I love that is genius. Yeah, there are a ton of things you can do to help clean up the lake. A lot of them have to do with preventing things from getting into the lake in the first place. So those are things like best management practices. So when you plant a garden, you want to make sure that you are containing all of the soils, so those soils don't, those fine particles don't run off with the rain down into the lake. There are ways that we can use fewer fertilizers. Like I talked about, those land plants need fertilizers. They need phosphorus and nitrogen to grow. But if we can use fewer fertilizers, then we can have less algae in our lake. We can also prevent microplastics, plastics from getting into the lake. So there are a lot of things you can do to stop them from getting into the lake in the first place. And then once they're there, that's where our scientific innovation comes into play. So I love your idea of creating a tiny robotic Daphnia. That's genius. So we are actually trying to, there's a plan, a research plan to remove some of these Meister Shrimp that are causing such, wreaking such great havoc on our lake. And by removing those Meister Shrimp, we are hoping that clarity will increase. And we actually saw a natural experiment happen. A while back, I think in 2012 or so, in Emerald Bay, which is down in the southwest quarter of the lake. Bailey is pointing to Emerald Bay there. At one point, we saw no Meister Shrimp in that part of the bay, and we aren't sure why, but when that happened, lake clarity doubled. So that's another way we're trying to help improve and protect Lake Tahoe.
And Heather just put in the chat that there has been discussion about using solar-powered boats to mechanically remove the Meister Shrimp from the lake.
Pretty cool.
Are there any other questions out there?
How do like the little vacuum repopulate?
How does what repopulate?
Like the Daphnia?
Yeah, the Daphnia.
How would they? How could they repopulate?
So as Elise was like mentioning, if we remove the Meister Shrimp that are preying, that are eating the Daphnia, then that's one way that we can bring the Daphnia back. But they also are asexual organisms, and they can just give birth to multiple Daphnia babies.
Clone themselves. I believe Heather said they could do either. Is that correct?
I think they can, yeah.
Yes, they can do both sexual and asexual reproduction. And so they can clone themselves, they can have eggs. The eggs can live in the bottom of the sediment. So as long as we could remove the population pressure, so if we could take away some of the Meister Shrimp that keep eating them, then the Daphnia populations can grow and grow and grow. And then the Daphnia can do the job that they used to do when they were native and, you know, when their populations were in high numbers. If we can bring the native Daphnia back by getting rid of some of the Meister Shrimp, taking them out.
Cool. Are there any other questions out there?
Anybody in Miss Scribe's class? Do you all have questions? Oh, you're all like, oh, I see one hand, or is that a shaking your head? No.
But other than that, we had a lot of fun sharing our science experiments with all of you today.
Oh, and thank you so much for coming and letting us record. It's been awesome.
All right, thanks you guys. This was great. Thank you so much.
Bye.
Bye.